Greetings, everybody. So last week, welcome to this one more seminar. So last week we had Susan Drazingaker from CIMIT Mexico presenting great research on wheat and how they are using molecular markers and molecular tools on their breeding program. So today we will be introducing maize research from Israel, Senegal, with the support of Dr. Vijay Shakam. So Dr. Vijay is a renowned scientist and managed the CIMIT double haploid production facility in Kiboko, Kenya, and conducts research on improving the efficiency of double haploid production process. So Vijay current research involves developing improved haploid inducers with high haploid induction rates, but also to introduce novel methods of haploid identification and efficient protocols for chromosomal doubling. So he works closely not only with the breeders to develop ways to using the DH lines more efficiently in maize breeding programs, but also contribute to CIMIT's work by delivering more than 200,000 DH lines and showing the enormous success of CIMIT and the maize team. So thank you very much, Vijay, for this opportunity, for your time. We are very honored with your presence, and we will let you add more to my words and present later, Georgina, to the audience. Thank you.
Sure, Anna. Thank you very much, Anna, for the nice introduction and for inviting me to make this presentation. So I briefly talk on the doubled haploid technology and what we are doing at CIMIT. Somehow it's not moving to the next slide. Yes. Now, so for productive and agronomically superior hybrids, we need pure breeding lines or genetically homozygous inbred lines. Anna, can you confirm you are hearing me well?
Yes, and right. We are seeing. Thank you.
We are saying, okay. So conventionally, the breeders use this recurrent self-pollination method, as you see in the top picture. So it takes almost 8 generations to develop these inbred lines, but the final inbred line is not 100% homozygous. It may be around 99% homozygous, but still there may be 300 genes. Even if 1% heterozygosity is there, there may be 300 genes segregating, which may not be desirable in breeding process. So, and as an alternative to this recurrent selfing method, from 1990s, there is another method called double haploid technology, which evolved from there. And using this method, we can produce pure breeding lines in just 2 generations. In the first generation, we produce haploids, and then we double the chromosomes in these haploids to produce the doubled haploids. These doubled haploids are 100% homozygous. And as you can see, the distinct advantage is it takes much less time, and the resulting inbred lines are 100% homozygous, okay? So the process of DH line production in maize, we use special genetic stocks called haploid inducers. These have some reproductive abnormalities in the pollen. So when you use that pollen onto any germplasm from which we want to derive inbred lines, it can produce haploids about 10% frequency currently. So these haploids can be identified using an anthocyanin marker at the seed level. So diploids have color marker on the both endosperm as well as embryo, but haploids will have color marker only on the endosperm but not on the embryo. So that's how we separate these haploids. Once we separate these haploids, we germinate this haploid seed, and we subjected them to a chromosomal doubling treatment using some chemicals that inhibit mitosis. So like a colchicine can help in this process. And after that, seedlings are recovered in the greenhouse and they are transplanted, and we give the best possible agronomic conditions so that these haploids can survive and they can produce some seed. These haploids are very weak because they have only one set of genome there, so they're very weak. So we have to give best agronomical conditions, and if only some of these haploids become doubled and they produce pollen, when you self-pollinate them, we produce these DH lines, seed for the DH lines. This is the DH process. This is in very brief, okay? And what CIMMYT had done over last 15 years is CIMMYT established several facilities, 3 different facilities. The first facility was in Mexico, and it was established in 2011. And then another facility was established at Kiboko, Kenya in 2012 and 13. And in India, recently we established another facility in 2021. So these 3 facilities serve not just CIMMYT, but also the NARS partners as well as the small and medium sized companies. So through these facilities, as you can see, we produce 304,000 DH lines from 2,843 populations. This is just from 2017. Okay, we started these DH operations in 2011-12. So we produced much more DH lines, and we served more populations. And as you can see, 63% of these DH lines went to CIMMYT maize breeding programs, 17% went to NARS breeding programs, and 16% to the small and medium-sized companies. So these facilities not only serving CIMMYT, but also CIMMYT as well as IITA, but also the NARS partners and private sector partners. In addition to these facilities, at CIMMYT, we also did a lot of research to improve this DH process specifically for the tropical regions. The first improvement was in terms of developing these tropicalized haploid inducers. When we use these temperate inducers in tropical conditions, they do perform very poorly. They're susceptible to all tropical diseases, and they don't even produce seed. So that's where CIMMYT collaborated with University of Hohenheim, Germany, and we produced this first generation tail hybrids were released. And considering the scope for improvement, we further developed the second generation tail hybrids. Currently, these tails were distributed to more than 80 different organizations, some mostly private companies as well as some public sector organizations. And in addition, there are several issues with haploid identification. So we developed inducers with red root markers, and we also developed other ways to identify haploids from diploids when these other markers do not work. And we also improved the efficiency of colchicine-based chromosomal doubling. So together, as you can see here, we did very significant improvements in terms of haploid induction rate. Initially, it used to be 7%. Now we have 12%. False positive, what we think as haploid, but it turns out to be diploid, we have reduced that very significantly to 3%. And the doubling efficiency has gone up from 6% to 27%. So together, all these improvements resulted in very significant cost saving. Now, per a population, it cost only about 2,200 US dollars, where we produce at least 100 lines or more. Previously, it used to, for the same, it used to cost us 3,000 US dollars. So together, we did not only establish the facilities, but also we did a lot of research that improved the efficiency of DH line production. And we published a lot of these publications, which can be accessed by all the breeding teams, and they can use these protocols. So coming to the last slide of DH user advantages of using DH lines. So this DH offers the quickest route to reach homozygosity. So it reduces the time spent in inbreeding, which in turn results in increased efficiency of breeding operations. For example, imagine instead of dealing with hundreds of thousands of F2s, F3 families, so you can just limit per population 100 to 200 DH lines, which is much easier to ship or from one location to another location, much easier to plant, and manage the nurseries, etc. So it improves the efficiency of breeding operations, and breeders can focus on identifying the best inbred lines and the best hybrid combinations rather than focusing on this time consuming and the resource intensive inbreeding process. So, and also because these are 100% homozygous lines, the use of these DH lines increases the correlation between phenotypic and genotypic correlations. So, and also these DH lines are very amenable to molecular marker applications like genomic selection or QTL mapping applications. So because of their 100% homozygosity. So now I will introduce our next speaker who uses DH lines for QTL mapping, and she will demonstrate how these DH lines are used. So coming to the next speaker, we have George. Let me stop sharing and I will introduce our next speaker. Our next speaker is Georgiana Ehemba. She's from Senegal, and she completed her bachelor's degree at Cheikh Anta Diop University of Dakar in 2014. And in 2017, she also finished master's degree in the same university in plant pathology and crop protection. During her master's degree, she focused on identifying downy mildew resistant pearl millet lines, which are currently used in Senegal breeding programs. And in 2018, Georgiana got a fellowship or scholarship by European Union to pursue PhD in plant breeding at WASI, that West Africa Center for Crop Improvement, the University of Ghana. She currently focuses on developing hybrids suitable for drought and low nitrogen soils in West Africa during her PhD studies. I think Georgiana is also currently working in the National Agriculture Research Program in Senegal, and she contributes to advancement of agriculture research and crop improvement efforts in Senegal. So with that, I request Georgiana, let me stop sharing. 1 minute. Yes, Georgiana, I think you can start your presentation.
Thank you so much. I am Georgina from Senegal, and I'll be presenting a work that I did. I was in genetic diversity and identification of QTL for low nitrogen tolerance in double haploid maize line through the mid-density genotyping. Well, okay. So this is the outline of my presentation. So maize is an important crop cereal and is a diploid, and it is the first most important cereal in sub-Saharan Africa with a high genetic diversity and more than 32,000 genes. It has a lot of values. We have nutritional values, and also is used as food and feed. So we can see that in West Africa, Senegal is at the 9th position with a production of 787,750 tons per hectare, which is a kind of low compared to other countries in West Africa. So there is a need to improve the maize productivity in Senegal. And then it is the third most important cereal in Senegal in terms of production after rice and pearl millet. However, this low productivity in Senegal is also coupled by by some constraints which are biotics for some and then abiotics. So here the focus of my work was on drought and low nitrogen, but the work that was done on genetic diversity was focused on low nitrogen because there were not much publications that focused more on the low nitrogen, so it was most on drought. So I wanted to focus the work on low nitrogen to see if we can find QTLs that can help in selecting lines under low nitrogen conditions. So when a plant is subjected to low nitrogen, the yield loss can go up to 50%, which is a kind of serious. So there's a need to focus on these traces. So we also have a low productivity of maize in Senegal. It's around 2 tons per hectare. And also the importation of maize to meet the food and feed demands is high. So there is a lack of inbred line, also in hybrids with low nitrogen tolerance in Senegal. Therefore, it's important to understand the genetic diversity among the newly developed double haploid lines to inform decision-making in our breeding program here in Senegal. And also we know that SNPs through mid-density genotyping have been used to determine the genetic diversity among double haploid line as well. So we also need to map the genes that will help improve the population through the marker-assisted selection. However, the general objective of this study was to develop high yield in maize, hybrid tolerance for drought and low nitrogen, for low nitrogen tolerance, sorry. So the specific objective of this study was to determine the genetic diversity and the population structure among the double haploid line, and also identify candidate genes that are associated with the low nitrogen tolerance. So what we did, we selected three OPVs that had been the most used here in Senegal, and then 2 in the WACI program where I did a PhD. Then we sent them to Vijay, who just concluded his presentation for double haploid development. So 100 kernels of each population was sent to them, and then they developed the line for us. After the line was sent back to us, based on the availability of the seeds, we chose 250 double haploid line and then did this study. 10 days old seedlings were actually collected and sent to Australia for DNA extraction using the DAT-SEC mid-density genotyping. Tassel was also used to map the DAT-SEC regimes, and with a minor allele frequency of 5%, a core rate of 80%. And then after filtering, we got 2,135 SNPs. So the mid-density genotyping actually used 3,305 SNPs in these studies. The data were analyzed using our computing, and then to compute the map, the minor allele frequency, polymorphism information content, the genetic distance, and the kinship matrix. The population structure among the double haploid line were assessed based on admixture analysis in layer package. And then to further explore the structure of the population, a discriminant analysis of principal component in AgeNet was done following Jumba et al. 2010. The candidate gene discovery were identified via the maize database genome browser with the B73 reference genome assembly version 3. The putative candidate annotation were retrieved via NCBI, which is available in this link. Google was also used to complete the information given by the NCBI. For the association mapping, we use GAPIT, and we perform the market trait association analysis. The blue best linear unbiased estimated was also computed using LMR in R. Manhattan plot was created using R, and a threshold of 3.3 was adopted to identify significant associations in this study. So from the results, we got a summary statistic which shows a mean minor allele frequency of 0.2, and then the genetic distance mean was 0.38. The polymorphism information content was actually 0.38. So some study have reported polymorphism information content which were lower than this polymorphism information content here, which shows that the SNPs used here were able to capture the variation among our SNPs. And also based on the genetic distance, we can see that the line had an acceptable distance among them. They were not really related. So here we have the SNPs density plot, which we presented in this figure. From the genetic distance, we also have a frequency distribution of the pairwise genetic distance, which we separated. Actually, what I did, I separated the yellow from the white to see how distant they are among them. So what I did, I used the pairwise genetic, Euclidean pairwise genetic distance, and then I classified them. So after classifying them, I realized that both the yellow and the white DH line, actually 80% of them has a genetic distance which were greater than 0.35. This shows how the lines were distant among them, and then we can proceed into doing other study using these lines. And then here we have the heat map of the SNPs among the 250 double haploid that we use. And then the color display here are also the distribution of the SNPs. Then we can see that the more the red here in the matrix, the more the color is brighter, the more related are our lines. So I went on and then I did a discriminant principal analysis, discriminant analysis of principal components. And the Bayesian information content showed that we had 5 clusters, which are here. So the variation here was shown by 4 discriminant, which shows 77% of the variation. So we had 5 clusters, and the line were classified based on their country origins and also based on their colors. So we realize here that from cluster 1, the cluster 1 and then cluster 2 had mostly lines that were from Senegal. But I can say that the cluster 2 actually had lines with the yellow endosperm colors. And then from cluster 3 to cluster 5, we had lines from Ghana, which also clustered together. But in this clustering tool, we could see some of the lines which belong to the Ghana group with the white color join the groups from Senegal, which had the white endosperm color. Then we concluded that after research, we realized that Obatampa that's actually grown in Senegal was initially taken from Ghana. So they may still be sharing some genes, or they may still have some kind of relatedness among them. And then I went ahead and did a diversity within a discriminant analysis of principal components within the white lines, which were from 3 populations. And then we also had 3 discriminants here. And then the line also fall into each population they were developed from. For the yellow one, we also did a discriminant analysis on principal components to see how related they are. And then we also realized that we had 2 discriminants, but here, only 1 discriminant were able to actually explain the variation among them. And we did a clustering and see which of the lines belongs to population, to cluster 1 and cluster 2. Then we realized that also most of them, even though there was some mixture in some of them, but then most of them also fall in the same population they were developed from. So here we have the admixture analysis, which also show us 5 subpopulations. Here, you can see the color green, blue, ash, yellow, and then red. From these ones also, the lines were actually clustered in the population they were developed from, with some of them having, with 3% of the line also, 3% of the line were actually in admixture. So like I explained earlier, some of the line were actually open by, some of the line were actually developed from Obatampa, which is initially taken from Ghana. So this could be why the line had some admixture, some genes sharing together. So that's the conclusion I can explain here from the admixture analysis here. Right. So somehow I'm not able to move the screen. Let me see. Okay, so from the association table, we identified 12 significant genes here, which are related to plant aspect. Some are related to yield, antecisoking, interval, and then some are related to stay green. So we went ahead and do some Manhattan plot here. Then we realized that from the Manhattan plot, we saw 2 genes that were related here to antecisoking interval and then the yield. From the antecisoking interval here, we had, first of all, we had this GRM2 G518198. These genes has been reported in Arabidopsis being able to help the plant fight against the biotic and abiotic stresses. It was also reported in drought tolerance on maize. So this could be the reason why it was found here in these studies, because some of the study of Bedu Apraku and al. In 2021, he reported that selecting for drought can be an indirect selection for low nitrogen tolerance. So because this gene were actually reported in drought, it could be also why it's helped the plant to also tolerate the low nitrogen here. So let me also, how do I even say? Let me say something also. From this study I did an agronomic performance of the lines. So from the agronomic performance of the lines I did, which were not represented here, all the lines that had these genes were tolerant to low nitrogen. They were tolerant to low nitrogen. And then the one here that was reported in the yield, that was found in the yield in chromosome 1, presented here, was a gene that was reported earlier being involved in the photosynthetic of the lines, helping plants also to close their stomata when there is a stress. And this is a process that's actually related to the flowering stage. That is a major part that these stresses affect. So it could be why it was found in a yield to help plants also tolerate the stresses. We found also other genes in antecisoking interval and then in plant aspect. In chromosome 2, we had a GRMZ2. Here, the gene is an S-type ion gene that was also found and reported being helping also plants to tolerate intolerance of some of them diseases and some of them in biotic stresses, which it was not really confirmed with biotic stresses, but it was reported helping the plants by also making some signal in their patterns when they senses any stresses in their plant, like when plant senses any stresses. The plant aspect here also gene were found here in chromosome 2. The gene was found in plant aspect. This is the GRM ZM2 G15 17 17. So this one was actually related to protein production under stress environments. And then it was also reported helping plants to produce more chlorophyll when being under stress. These two genes were reported under stay greens, the genes which are GR, MZ, M2, G3, 52, 891, and GR, MZ, M2, G0, 1761. Both were reported in stay green stages. So they actually are genes that were reported in helping the plant to maintain their, they actually played a role in the silking, in helping the plant be staying green, but also were reported helping the plant in the production of silk in maize. And then the last gene was also found in yield and mostly related to the protein production, and it was reported previously being able to help plants maintain their productivity and it was reported in some crops like rice and what's the name of the other? Sorry, it was reported in rice, and there was another crop that escaping my mind. So that's the last one we found in these studies. So in conclusion, we said we had a high genetic diversity and a population structure within the GH line. And then 5 subgroup and 5 clusters were obtained in these studies, both by the admixture and the discriminant analysis of principal components. 12 markers here were found associated to stay green, yield, anti-silking interval, and plant aspect. Then the candidate gene functional annotation revealed that most of them are involved in the photosynthesis. Almost all of them had a way to play, one way or the other to play in the photosynthesis, which help plants to stay green and also produce enough chlorophyll, and some produce protein, and some to also tolerate, to help tolerate the low nitrogen stress. So these genes that were mapped here mostly are involved in biotic and abiotic strains. And we can say that this gene can be used to help in the selection of low nitrogen stresses. So I'd like to thank the Senegalese Institute of Agricultural Research here where I currently work. I'd like also to thank WACI, where I was when doing the PhD, and thank CIMIT, who funded part of this work. And thank you, Vijay, for that Hub development line. Thank you so much.
Thank you, Georgina, for the excellent presentation. So we open now the floor for questions. So we have already one question from Dr. Gary. Do you want to ask directly to Georgina, please? You can unmute us yourself.
Okay, please. So should I stop sharing?
Yes, you can stop sharing, and Dr. Gary can unmute himself.
Okay. What is the question?
So the question from Dr. Gary was the phenotyping done on the lines per se or on the test crosses?
It was done on the lines.
Okay. So I don't know, just raise your hand if you have any question. I have one question to Vijay and one for Georgina. So for Vijay is how can we support more CIMIT partners with the DH lines? And how can they ask support? So can you explain briefly the process, Vijay, please?
Yes, Anna. Currently, most of the NARS partners in Africa are being supported through projects like AGG. We have allocations. For example, in Southern Africa as well as Eastern Africa, all the partners are able to get 10 populations free of cost. So they can send me the details, and within a year to year and a half, they can get the DH lines after plant in the induction nursery. So, but many times some of the partners, they may have their own projects and which can support this type of DH service. And then they can also request like that. Currently, the service charge is $2,200. It's a cost recovery charge. It's not profit making. So if per population, we produce at least 100 lines. On average, we at least tend to give out more than 150 DH lines. Okay.
Thank you, Vijay. So Georgina, my questions to you is, how can CIMMYT support more your work on maize? This is the first. The second is, will you continue in Senegal to research on maize? And what is the next step for this work? Because many congratulations, you finished, I think, last week your PhD. So I would like to listen a little bit more about it. Thank you.
Thank you, Anna. Yeah, so CIMMYT can support actually my work by maybe collaboration. And if there is also available projects that they are doing there and think that we can do it here in Senegal, they can also support and we do it together. Yeah, and please, the second one was I'm currently working on on the maize breeding program. I'm actually in charge of the maize breeding program in Senegal, which I already started last year at the rainy season. And actually, I multiplied some of the DH line last year for hybrid development. So actually, and then also this dry season, I have set experimentation to make crosses so that I can get hybrids and continue in the future. Yeah, I just defended my PhD and I want to continue in the research area with the maize so that we can boost the maize productivity here in Senegal. Yeah, thank you.
So what it will be the continuation of this work?
So from this work, I would like to validate the QTL I identified here and then use it to fasten the breeding program here. And also from here, I will be focusing on producing more hybrids for farmers use here. And also here is a lot of maize for feed. And then we can also help the industry here in Senegal. Thank you.
Thank you. So I don't see any question or any hand raised. So if there is, so maybe Mokhtar, do you want to unmute yourself and make the comment? So Mokhtar is saying, thanks, Georgina. DART offers DART-seq HD and LD. Is the mid-density referring to the DART-tag targeted genotyping? So maybe you can answer this question.
Yeah, so yeah, actually we use the how do I even say it?
DART-tag.
Yes, that's take mid-density genotyping to do this work.
So maybe his question or his doubt is that why you didn't use the DART-seq high density or low density, since it have more markers.
Oh, okay. So I was actually offered the mid-density genotyping and the support I got was the mid-density genotyping. Yeah.
Yeah, thank you.
So it was offered by us by Anna and Co. Thank you so much, Anna, and your team. We are so grateful.
Thank you, Georgina. So Mokhtar also make a comment, is that if you can make any comment regarding the gaps in the physical map in relation to the number of the significant SNPs identified and the confidence interval of the identified SNPs. In other words, do you think the SNP panel is adequate or suitable for the discovery studies?
So sorry, I was muted. I was saying yes, I think, but I didn't know I was muted.
So I think, Mokhtar, like the partner said, I think one of the major problems with the NARS is really the financial support. And what it was offered was really the mid-density dark tech. Definitely, I think all the studies in relation to discovery, they have the advantage if they have utilized the high density number of markers. However, this is not possible for other partners, but suggestions from everybody they are very welcome. Bodo, do you want to unmute yourself and make the question?
Yes, okay. Hello, Anna. Hello, everybody. Thank you. Yeah, I found this very interesting, and it is really appealing to me how this DH work can facilitate maize breeding nowadays so much. And I was quite surprised because this description given by Vijay akins a lot with description of how to make haploids of potatoes. So I know from the potatoes, as I worked with it for a long time, that everybody who gets a hold on such a pollinator with the anthocyanin marker can carry out this kind of haploid production by themselves. So that is why my question would be, CIMMYT has now these central spots where we carry out this DH production, and we are also supporting nationals. Is it recommendable to stay with these centralized facilities, or can and is already this technology be carried out at many different points across Africa at least? Thank you.
Okay, let me answer that question. So CIMMYT, it took us almost at least 7-8 years to reach at this level of efficiency. So there is a big learning curve within this DH. Maybe I made it too simple, but there is a big learning curve for this to adapt this technology. Second thing, the limitation is the haploid inducers. They are not easily available, and now the people can get from CIMMYT. But if they don't manage these haploid inducers properly by regularly testing this haploid induction rate, it can quickly deteriorate, and within 3-4 years the haploid induction rate can come almost to 0. So that is the technology need to be refined. So another thing is doubling these chromosomes. It involves very toxic chemicals, so we need to have proper facilities, lab and disposal facilities, so which are establishing these facilities are also costly. And if the breeding programs do not have enough scale, for example, if you just need 200-300 lines or 1000 DH lines, you don't need to establish these big facilities which are needed. So in that way, the centralized facilities offer service at a much bigger scale and much more efficient at lower cost. So that is the idea for to establish these centralized DH facilities. And again, as I told, several industries as well as NARS partners, they got our haploid inducers, and many of them have established their DH in-house DH line production. So it's possible to do that. Only thing, if you are able to operate at scale, then it will be cheaper and efficient.
Thank you so much.
Okay.
Gary, you raised your hand. Please unmute yourself and you can ask directly the question, please.
I think Joseph was first. Is he not?
Yeah, I think, okay. So, yeah, mine is, okay, congratulations for your nice presentation and the study in Senegal. Yeah, I think one point is in terms of that QTL, the population, I think the grouping of the population is based on the cross they derived. I think that is expected. So mine is just if you focus on product development, you will be successful rather than targeting individual QTL, which is very difficult for drought and low nitrogen. So just mine is just a comment. Thank you.
Thank you.
Gary?
Yeah, I guess I've got basically the same comment. Congratulations on the work and on your program. I would like to step back a little bit and address a comment to CIMMYT scientists and I guess wacky professors who about how we design these PhD theses and how we allocate resources, in terms of DH, for example, or other research resources. So that this was a well done study, but there's two basic problems. One is the problem that Joseph raised, is that it's very unlikely that we will find usable haplotypes that you would say want to include in an index or introgress into target germplasm for a trait like yield under low N. And it's especially unlikely that you would find it in inbreds per se, rather than in test crosses. But either way, there's not really a pathway forward now for this. So my suggestion would be that in the formulation of thesis projects, and when CIMMYT is presented with a request for DH to support a thesis project, that you engage on the actual project, so that we can make sure that this effort, ties into a practical outcome that can move maize breeding or plant breeding in the region forward, and not just sort of be a good thesis project. We need to separate this idea, or try to. There are many good thesis projects we can do that are that will contribute directly to problems that are solvable, and that will help move the breeding programs forward. So, and to identify those projects, it's important that the researchers, the students, the academic advisors consult together on what is the best use of time and resources, and the students' hard work. So congratulations again on completing and on this big step.
Thank you, Gary. So I have a question for you. My question is, I agree with you that CIMMYT should be involved since the beginning in the projects, but how to surpass that gap means, instead of the partners come to us just almost in the end of the project, how to engage them more since the beginning, if you can help us on that. Thank you.
Well, I mean, the project used CIMMYT DH, so they had to request those doubled haploids, right? So that's the moment to have that discussion. What are you planning to do with these DH? It seems to me, if CIMMYT is going to take a student on or provide resources, so to generate 200 DH is it's not a huge amount of investment, but it's a considerable amount of investment.
But it is taken
Then
as a
that's the
service.
point to discuss it at, I would think.
Gary, but if this is taken as a service, and that is one of the issues when we transform research into services, it's not a critic, it's just a point. How can we engage more debt partners to don't be so individualized, but really to collaborate with us and sharing their plans? Because when we are a service, the service is requested, and we have to provide that service. This is also serving for genotyping, and any advice is most welcome.
Yeah, just quickly, a service doesn't imply that, especially in this context, you are not a commercial service, right? You're not getting full cost recovery either for genotyping or for DH, and the funds with which these are, the request isn't coming from a commercial enterprise. We are all part of a public enterprise to deliver improved maize varieties to smallholder farmers in sub-Saharan Africa. So, and the capacity of the CIMMYT DH facility is not unlimited. Or genotyping capacity, more or less, I think, is unlimited, as long as somebody can pay for genotyping from one of the commercial service providers. Then that's fine. But I would still argue that research resources, the grant money that is driving these projects, permitting these projects, is very limited. So it's reasonable that we're essentially looking at a rationed resource, a resource that is scarce. The CIMMYT, I think would have difficulty generating many more DH lines than it currently does annually. So there's a rationale there for allocating those resources to projects that are going to have the greatest impact. So it's not to say CIMMYT necessarily should have a veto, but should feed in scientifically to the design of these projects. And that, that will help the student. It will help the research programs, should help everybody.
Yeah, thank you. Dorcas, do you want to unmute yourself and ask the question directly, please?
No, it's not a question. Thank you, Anna. Thank you for the presenters today. It's just a comment following up on your question about being a service provider. And I think we had started a discussion on this previously. And one of those things should be, for example, the services can be asked to be provided if they are genotyping or they are DH or whatever, to be linked to an active breeding pipeline so that at least those materials are moving forward in the breeding. Yeah.
Thank you. And Georgina, this is an advice for you, so maybe you also can follow up with Dorcas later on. So I
Yes.
have a question for Cesar. I agree with you that for discovery, we really should use higher density of markers because the probability also to discover is bigger. But should you think that we should change the description of the mid-density panels that are tagged in our website? And because one of the things that we say is that it can be for studying the structure population. Should we change that, Cesar, or can you give an insight about it, please?
Yes, thank you, Anna. I believe that, for example, in the previous discussion, just some minutes before, Mokhtar was asking about the discovery studies. I believe that DART tag is not recommendable for discovery studies because you have a marker panel specific for some traits. And to me, I suggest to use, for example, DART-seq for discovery studies, even the high, mid, or even the low density option. In terms of if you need to change right now what you are using, I guess that it is possible, or it is an option to change it, but we need to try to discuss about that and be more specific in terms of what you want first. So yes, if you want, at any moment we can have a meeting and discuss about it.
Thank you. So Mokhtar, maybe you can unmute yourself and say, because I think this is very important for partners to understand what is possible and what is not possible, and to don't have contradictory information. So Mokhtar, do you want to unmute yourself? Otherwise, I can read particularly the second part.
Thanks, Anna, and thanks, Georgina. Actually, it's just a follow-up comment on Cesar's comment. And I was just saying that in general, when we create dot tag panels, it's mainly for GS because, we kind of filter the markers based on their minor allele frequency and other factors. But then to reduce, let's say, the density, we even exclude a lot of markers that actually have a good minor allele frequency. So when you do discovery, and also Georgina showed it in the physical map that there was a lot of gaps. So if there is a gene lying within those gaps, the discovery study may fail to identify them. Identify the linked markers. So actually, it's just a comment to consider for next studies. But it was really interesting to find if they found some significant markers, even with this low density, it's worthwhile actually really validating them and maybe see if there is a way to either develop CASP markers or doing a fine mapping to really have very significant CASP markers that they can use in the breeding program.
Thank you, Mokhtar.
Thank you so much.
Stelio, please unmute yourself and make the question. We are not listening you. Stelio? We see you unmuted, but we don't listen you. We don't hear you. Not very well. Maybe you can speak louder. Otherwise, you can type your question in the chat and we convey to Georgina and to Vijay. Also to know that and to emphasize that you can reach Georgina and also Vijay for any question, any particular question, or even to us, and we also will deliver the question to the presenters. So if there is no more questions, I think we can end the session here. So thank you very much to all for this presence. Thank you, Georgina, for the excellent presentation and work. Thank you, Vijay. Thank you very much. We hope to see you next week for our next seminar focused on sweet potatoes. And this will be presented by Dr. Godwill Makunde from Mozambique and Ruben Sali from Uganda, both from our esteemed partner center, SIP. So both will be introduced by the renowned colleague, Dr. David Decoy from the AAFFC from Canada. Thank you very much and hope to see you next week. Thank you.
Thank you, Hannah.
Thank you, Anna, and bye.